morning, students. Today's topic is cell senescence and apoptosis. So, what do you mean by cell senescence? Cell senescence is nothing but aging of the cells. How the aging of the cells is going to happen, and what and all are the changes that we could see in the cell when the cell is going to become an aged one. Okay, and if we have, there are theories put forth for this how the aging is going to happen, and there are many theories. Many scientists have still doing the researches to find out how the senescence is going to take place. If possible, if we can prevent it, then of course our aging would not take place, and we can prevent our aging, and we can have our lifespan little more longer than that of what we have. Okay, that is only the ultimate goal. So today we are studying the cell senescence. So what do you mean by cell senescence? It is nothing but it is a deteriorative process which naturally slows down and terminates the functional life of the cell. So the functional life of the cell is going to decline, and further there is no function of the cell, which ultimately leads to the death of the cell. Okay, so then the cell senescence, how is it going to actually happen? The organizal senescence is going to take place, and further there is a death of the cell. Suddenly the cell cannot die off, isn't it? There is some organized method for which the cell is going to get senescence and it is going to die. The cell and organisms are not immortal. So of course, if the cell is not immortal, of course the organism is also not immortal, isn't it? So both are not immortal, which allows us to study today's topic, that is cell senescence. Cell senescence is indicated by what and all methods. So it is if there is a decline in the functional efficiency of the specialized cells, like the nerve cells and the muscle cells, which are not involved in the divisions. Okay, if their capacity of the functioning is getting reduced, then the cell is going to be senescent. Thus, the decline of the division capacity. If there are the cells which are continuously dividing in our body for the repair, for the growth, all the other purposes, if those cells are declining in their cell divisions, okay, replicative senescence we call it as other words. So thereby the activity of the dividing cells reduces, and henceforth leading to the senescence. This examples is the lymphocytes and the epithelial cells. Here are the few cellular changes that we have listed out. Morphological changes that we would see when the cell is getting senesced. Then the change in the cell size and shape, atrophy we call it as. Then accumulation of lipofuscin pigment, which is going to ultimately lead to the uh, death of the cell. Then the nuclear penosis. Then you have lipid vacuole formation. Thereby, if these and all changes are these and all are the changes during the senescence. The physiological changes. What and all are the physiological changes that is going to happen during the senescence period? Accumulation of the calcium ions are going to take place. Loss of ribosomal RNA, decline in the transcription and the translation. Translation is nothing but the protein synthesis. Then stiffening of the glycogen, decline in the energy production. If there is, how does the decline in the energy production takes place? When there is a destruction or there is a decline in the processes of the mitochondria and the ribosomes, which in turn will lead to the translation, that is protein synthesis is going to decline, and also there is an energy production decline in the body, in the cell. Okay, the subcellular changes that we could see here is the change in the fluidity and the permeability of the plasma membrane. What in any molecules that is there which is nearby the cell, which will enter into the cell, or which any molecules which are there inside the cell is just being let out without the signals of the cell. Okay, without any permission, they are just left out or let inside the cell. The permeability reduces. Decrease in the granular endoplasmic reticulum, degeneration of the mitochondria, which I discussed a little earlier, that is decline in the energy production, which is in turn being connected with the degeneration of the mitochondria, degeneration of the chloroplast in case of the plant cells. So this is the fibroblast cells which you can see here, and this is the senescent. fibroblast cells wherein their shape and sizes being completely changed. Theories of senescence. Why are we going to study theories of senescence today? See, there are many scientists who have put forth what is the mechanism of the senescence. Okay, so ultimately the cell has to die or the cell is getting aged. What is that making that cell aged? Is what is the question today, isn't it? So there are scientists who have put forth according to their idea and their visuals. They have found that there are six theories that we are going to discuss here today. What is that? A small disruption which is going to take place in the cell, which is making the cell senescence. The DNA damage theory. The first theory is the DNA damage theory, and I would like to tell you another thing. See, these experiments are done only on the animal cells to know how the animal cell is getting senesced. Okay, so the DNA damage theory, accumulation of the DNA or accumulation of the damage to the DNA inhibits the cell functions and the cell divisions, further leading to the death of the cell. So declining the DNA repair capacity, if there is any damage also, their cell has a capacity to repair it. Okay, if that process is also being declined, then there is an cell then there is an no the gene expression is inhibited, of course leading to the death of the cell. Now this point is linked to my previous video. There I have taught you all the cell cycle and its regulation, correct? There in that regulation we have told during the cell division, if there is any damage to the DNA, then the cell the cyclins and the CDKs, that is cyclin dependent kinases, are going to fix the cell, try to repair the damage of the cells. Okay, if that is not going to happen, then they are going to kill that cell. Okay, the same process here also it is going to apply. If there is any damage in the DNA, they will try to fix the problem first, repair it, but if that is also not possible, then the cell is led to the death. Okay, the decrease damage in the mitochondrial DNA and the decrease in the energy production. Okay, so then built-in broken down theory. What is built-in broken down theory? Is nothing but the genetic material when there is lost or when there is any damage in the genetic material. Of course, DNA is also a genetic material, but here what are they going to speak about is some certain genes are there which is going to express after a fixed lifespan of the cells. Okay, every every stage of our life do not have that particular same functional genes, okay, functioning. Particularly certain set of genes are going to perform or are going to function during a particular stage of a man. Okay, in a childhood there are certain set of genes which are going to actively participate. Then in adolescence there are certain genes which are going to actively participate, which is making the body look like a matured organism. And there are certain cells which are playing a very important role during the post maturation period, that is in the 40s and 60s, 40s to 60s age. Okay, after that there are certain cells of genes which are expressed to inhibit or the lifespan of the cell is going to decline. So thereby there is a built-in broken down theory what they are trying to explain you all here. Expression of the genes coding for the hydrolytic enzymes and the formation of the DNA synthesis inhibiting the proteins is also leading to the damage in the genetic material. Next is the telomere theory. What is this telomere theory says actually is, say when the cell is dividing, okay, once the cell gets divided, the telomere gets shortened due to some of the other reasons. Maybe the synapses is taking place, exchanging of the genetic materials within the chromosomes. Resulting in all this, there is a small, minute decrease in the length of the telomere in certain cells. Okay, thereby what is going to happen is, as the divisions is taking place, further divisions is happening and happening. The cell at one particular time, the telomere becomes very short, which is not capable of further division. okay, thereby resulting in the decline of the cell divisions. Once the cell division is stopped, then the replicative cell senescence is going to occur. Okay, so free radical damage theory. What is this free radical damage theory is actually about? A very important theory, if I could tell you. Today, the scientists are actually focused on this particular theory. Okay, very vast theory, very important theory, which says how exactly all these other theories are going to tell us, isn't it? The damage in the DNA, damage in the genetic material, shortening of the telomeres, all these are happening due to what reasons? Correct. So that answer is given by this particular theory, that is free radical theory. These free radicals which are there in our body, which are radioactively react they are highly reactive in them. Okay, so they are going to create certain problems. Likewise, disrupts the cells and the nuclear membranes, alters the membrane permeability, damage in the DNA and the protein synthesis. So, how to come about the radical damage? By increasing the antioxidants, which reduces the free radical damage. Now, our goal is to increase the antioxidants so that in turn it reduces the free radical free radicals in our body, which in turn will reduce the damage which is occurring by the free radicals. Okay, next is glycation theory. What is this glycation theory? Is nothing but there is a cross linkages between the glucose and the protein molecules, which stiffens the cells. Okay, once the cell has to be rigid, and it has to be intact to a particular place, and it should be stiffened. Okay, so if that has to happen, then these links are very important. Once this links breaks down, then there is no stiffening of the cells, thereby the cell becomes flaccid, which will in turn results in the senescence. Immune theory, antibodies against body's own cells, which will again result in the cell senescence, that is autoimmune response is actually initiated. Endocrine theory, wherein the pituitary gland is going to give the signals or triggers the senescence to the all to certain cells which are not to be, or which has to be undergo the senescence. Importance of understanding the senescence. How important it is to understand the senescence. Extending the lifespan of the cells is very, very important. That is what we are today, correct? So we are more focusing on how to live a longer life, correct? So here the extension of the lifespan of the cells and the organisms by delaying the senescence. That is why we are going to study the senescence. Understanding the formation of the cancerous cells, failure of the senescence mechanism. By failure of the mechanism of the senescence, the cells are going to lead to the cancerous. Then, the cure for this cancer is also by promoting the senescence. Once you promote the senescence to allow a process of the senescence completely, then the cell will not change itself to become a cancerous one. Okay, that I'll show you here in a small graph, wherein this ultimately shows how, see, the growing cell under the stress or the loss of suppression, the abnormal proliferation is aberrant cell proliferation is happening, thereby the senescent cells are seen. This accumulation of the senescent cells together, they result in the damage, they completely destroy of those cells. Okay, degeneration of those cells is going to take place. See, accumulation of the senescent cells will sometimes allow one particular cell or few more cells in that particular group will themselves do not undergo complete process of the senescence, which will ultimately result in the formation of the cancerous cells. Okay, that I'll show you here in this picture also very clearly. This picture is actually explaining us how exactly the senescence is happening and further leading to the tumor cells or the other cells. See, cell senescence is actually important for many other processes also. Not only aging, the cell has to get older enough, and it has to end its life. Okay, for there are certain advantages also in that, but there are so many disadvantages too. Okay, see, under the cell, normal cell or the tumor cell, under the stress, it is going to become a senescent cell, and thereby accumulation of the senescent cells are going to take place. Reprogramming is going to happen. If the reprogram is going to happen, it may further lead to the senescence and aging. If not, then it's going to further lead to the cancer, and sometimes it can occur even the pulmonary fibrosis also can occur. If the immune cell activation is happening, then there is a senescent clearance, senescent cell clearance is going to happen, thereby resulting in the tumor regression, tumor suppression, tumor homeostasis, rejuvenation, and embryonic development, and the regeneration is also going to occur in this particular processes. Okay, so here ends up the cell senescence. Now, what is apoptosis? Next picture, what is apoptosis here? See, that is one of the process which is getting aged and the cell is going to die. Okay, here is the process wherein the apoptosis is nothing but it's a programmed cell death. We are planning for the death of the cell. Previously, we are forming the uh, planning of the cell is made so that the cell can be programmed to death. Okay, so in other words, we can say it as cell committing suicide. Okay, so by the activation of the intracellular death programs, the cell is going to commit suicide. Okay, that is nothing but the programmed cell death or apoptosis. What do you mean by apoptosis? Apoptosis meaning falling off like leaves of the tree in an autumn season. Okay, a neat silent death of the cells. Okay, very silently there is a death of the cell which is taking place in the apoptosis. No damage to the neighboring cells. Likewise, that of the necrosis, cell death is going to occur by swelling and exploding due to the injure or damage. Then you can see there is an different no different from autophagy also. What is that? Intracellular digestion by acid hydrolysis of the lysosomes, that is not going to take place here. Then here mainly mediated mainly it is mediated by a group of proteolytic enzymes called as caspases. So here the caspases is going to result in the cascade reactions, thereby resulting in the programmed cell death. Okay, so here there is no certain lysosomes or any other picture, other cells are going to come into picture. Okay, like that of the autophagy or like that of the necrosis, nothing is going to happen. Here slightly there is a difference in the apoptosis. That is what we are trying to tell you here. Present in all cells in an inactive form called as procaspases. What is this caspases, or as I told you, there are the proteolytic enzymes called as caspases. These caspases are present in all the cells. Okay, once they are getting activated, then only the apoptosis is going to occur. Okay, they are present in all cells which are in inactive stage. Okay, so once they get activated, then they result in the apoptosis. Activated to kill cells by death signals. The poison pills of the cells. These caspases are nothing but the poison pills of the cells. In the other words, which you can ultimately understand it as a poison pills for the cells. Okay. Once they are activated, then they are lead to the death of the cell. How actually the process is going to happen? See here, a normal cell, the cell shrinkage is going to happen due to the chromatin condensation. Then membrane beveling is going to happen. Then the nuclear is getting collapsed and continuously beveling is going to take place. Then the apoptic body formation is happening. That means there is a destruction of the cell and making it into the smaller pieces, that is the apoptic bodies formation. Then the lysis is going to take place, thereby apoptosis bodies are completely destroyed there, okay, degenerated completely into a smaller pieces. Thereby these macromolecules are further they are recycled back and the other is left thrown out. What is the mechanism of the programmed cell death? Nothing but PCD or apoptosis. Here, the reception of death, reception of death, that is nothing but the signals who are recepting it, the signals of the apoptic signals, which might be the internal or it can be external. Internal is by nothing but the caspases which are there, which get activated and further there is a destruction, okay. External also, the apoptosis can take place once they are damaged, or once they get externally stimulus is given so that the cell can die off, okay. It can further lead to the internal environmental changes and then the cell can get apoptosis. See, sorry, activation of inactive procaspases in the cell to active caspases, resulting in the cascade reactions which is going to take place further, which is leading to the degeneration of the cytoplasmic and the nuclear proteins altogether. Fragmentation of the DNA is going to take place, the splitting or the breaking down of the DNA molecules and the nucleus, resulting in the shrinkage and the bubbling of the cells, okay. The formation of smaller membrane bound structures, that is called as apoptic bodies, which I showed you in the previous diagram here, thereby leading to the lysis of the cells, or this digestion of the apoptic bodies by the macrophages is going to take place, that is nothing but the phagocytosis. Recycling of the macromolecules is ultimately going to happen at the end, so that whatever the useful materials, if possible to be recycled, it can be recycled back to the process. Okay, this is the process of the apoptosis. The normal cell, see here the mild contractions of the nuclear cell membrane is going to take place, and the nuclear fragmentation is happening there, which you can see all the nuclear pieces is being found, and then the bubbling, meaning the complete breaking down of the cells, and the cell fragmentation is happening, the apoptic bodies, which are in turn taken by the phagocytic cells, okay. What is the significance of the programmed cell death? Why should we study the programmed cell death? Normal processes during the development responsible for sculpturing of the body parts during the development of the animal. Okay, so very important process. Your palm, which you see, which has proper sculpturing, the toes which are formed, that is due to the apoptosis which is taking place. The complete mass would have been there before, isn't it? It's just like a bud of tissue, but because of the apoptosis, you can see the cells are dyed here, and it is giving rise to a fingers. Metamorphosis of the tadpole into the frog, insect larvae into the pupa, and then further in becoming into an adult. Cells of the tadpole tail undergoes apoptosis during the metamorphosis into to become a frog, and then balancing the number of cells in developing tissues to maintain the homeostasis, adjusting the number of neuron cells or the nerve cells to match the number of the target cells. That is very important. During this process, about 85% of the certain cell population died during the development of the vertebrate nervous system, okay. So thereby, that is example is the dorsal root sensory ganglia of the chick embryo. If it has to develop, then 80% of the cell declination is taking place because of the formation of the nervous system. So how important the apoptosis is, you see. elimination of the unwanted, improperly formed or functionless dangerous cells, that is very important, which I have to throw out of the body. Where virus infected cells, cancerous cells, improper developed the cells and activated lymphocytes, all those has to be destroyed. Then you have next is the application of the cell, application of the apoptosis. How can you utilize this particular programmed cell death to our uses? Maybe if naturally it is happening, it's very good. Okay, if we artificially induce, what help it is going to give to us? Okay, say like killing the cancerous cells by activating the pdc, we are going to externally impose a pressure on a cell to undergo a programmed cell death in case of the cancerous cells, so that the cells itself dies off. Okay, so one of the important application that we could see in case of the apoptosis, certain autoimmune diseases also can be reduced, or it can be due to a failure of the apoptosis in cells which should die. Okay, the cells, if when it has to die, certain immune diseases are not going to occur. If this autoimmune system is proper, then the proper functioning of the cells can take place. If that is being failure, then again it is leading to the improper functioning of the cells, which in turn, or which is giving you the way to the failure of the apoptosis if the autoimmune system is not working. Apoptosis in cells which should not die leads to abnormal development disorders or the neurodegenerative problems. So the cells which are need to be died, it has to be died. Okay, otherwise they are going to ultimately create problems in the organism, not the beneficial one. Okay, here ends your topic, the applications and the processes, and also the a few of the significance of the PCD, that is programmed cell death. So in today's topic, we have studied about the cell senescence and the apoptosis. Okay, so here is the picture of the programmed cell death. The cell shrinkage, chromatin condensation is happening, which I have already drawn and shown you in the previous diagrams. This gives you an ultimate clear picture, which I showed you and discussed earlier. Okay, so if you have understood the both the topics, and if you have really liked it, then like the video and subscribe for it. Thank you.